SRI gene. Full form is sex-determining region of Y chromosome that responsible for testis-determining gene produces transcription factors, acts on Leydig cell and Sertoli cells. Leydig cell produces testosterone, Wolffian system stimulation, male organs development. Sertoli cells, anti-Mullerian hormone, inhibits female organs development. Absence of SRY gene makes a female. Puberty. Prepubertal, continuous release of gonadotropin-releasing hormone. Inhibits release of luteinizing hormones and follicle stimulating hormone. Continuous release maintained by GABA and melatonin. At puberty, adequate fat stores increase leptin, permissive role in puberty increases peptin, GPR54 in gonadotropin releasing hormone neurons, pulsatile release of gonadotropin releasing hormone. When gonadotropin releasing hormone drops, it stimulates production of luteinizing hormone and follicle stimulating hormone. Male reproduction, scrotum, glans penis, vas deferens, epididymis, seminal vesicle, and ureter. One scrotum is 1 to 2 degrees Celsius less than body temperature to favor spermatogenesis. Diagram of testis, epididymis, vas deferens. Uh, this is the cross sectional. Spermatogenesis location in the seminiferous tubules, duration 74 days, process type B, spermatogonium stem cell mitosis, 2N primary spermatocytes, meiosis 1, reductional division, secondary spermatocytes 1N, 1N, meiosis 2, mix 4 cells formatted, undergoes differentiation. Meiosis 2 is the reduction, meiosis 1 is the reductional division. Differential spermatogenesis makes sperm and then release spermiation. Release is called spermiation. Cells in testis, functions of Sertoli cell. These are the cells in testis. Leydig cell produces testosterone, interstitial cells, myoid cells, spermatogonia, primary spermatocytes, secondary spermatocytes, round spermatids, spermatozoa, Sertoli cells. Functions of Sertoli cell, production of anti-Mullerian hormone AMH. Sertoli cell produces anti-Mullerian hormone, reduction of female organ development, blood testis barrier, tight junctions between two adjacent Sertoli cells. Blood testis barrier is due to tight junction between two adjacent Sertoli cells. Nerve cell of testis provide nourishment and production to sperm with the Sertoli cells. Production of androgen binding protein ABP, which binds to testosterone aromatase, causes testosterone to become estrogen. Inhibins that inhibits follicle stimulating hormone, phagocytic function, ingestion of dead sperm. Semen contains sperm plus fluid produced by seminal vesicles, 60%, prostate 30%, vas deferens 10%, pH 7.2 to 7.4, sperm count 90 to 120 million per milliliter. Sperm lifespan 1 to 2 days following ejaculation. Sperm maturation, acquirement of motility in epididymis, increase forward motility by calcium channel gaps for maturation of ectrodome. Note, vaginal secretion increase sperm motility. Length of a sperm is 55 to 60 micron. Head and tail. Tail velocity 1 to 4 mm per minute. These are the microtubules in the tail, which is called flagellum. Ecrosome contains hyaluronidase and proteolytic enzymes required for penetration of zona pellucidare. Surface membrane, vacuole nucleus, neck, body, mitochondria, and indel piece generates ATP for motility. Sperm capacitation. After ejaculation into female genital tract, ectoderm reaction, release of enzymes for penetration of zona pellucidare, the fertilization. Fluid component produced by semen is equal 60%. What does it produce? Fructose is the energy source. GLUT5 mediated energy source, fructose. Ascorbic acid, vitamin C is the antioxidant. Prostaglandin makes the cervical mucus penetrable by sperm. Phosphatidylcholine detected via Florence test for seminal stain in red traces. The chemical we detect by Florence test is phosphatidylcholine. Buffers which are used in this test are bicarb and phosphate. Sorry, not in this test, but the buffers in the fluid are bicarb and phosphate. Reduced by prostate 30%. Phosphates produce fibrinolysin, liquefaction of semen in 15 to 30 minutes. Liquefaction of semen in 15 to 30 minutes. Fibrinolysin acid phosphatase. Spermine detected via Barberio's test. Spermine detected via Barberio's test. Vas deferens 10%. Bicarb and phosphate is, is secreted by vas deferens 10%. Male sexual act. Reduction of penis facilitated by parasympathetic, that is sacral 2, 3, and 4 roots, mediated by acetylcholine, non-adrenergic, non-cholinergic system, non-adrenergic, non-cholinergic system, nitric oxide, dilatation of arterioles, compression of outflow veins, engorgement and erection, acts via cyclic GMP. Applied aspect. Cyclic GMP degraded by phosphodiesterase. Phosphodiesterase inhibitor, sildenafil. Increased cyclic GMP, increased nitric oxide, increased erections. Phosphodiesterase inhibitor. Cyclic GMP is secreted by phosphodiesterase. Ejaculation. Sympathetic T12 to L2, now roots thoracic 12 to lumbar 1 and lumbar 2, now roots 2 steps, emission, movement of semen into urethra, and then ejaculation, release of semen from urethra. Dissolution, detumescence, D2, detumescence, endothelin, and noradrenal, constriction of arterioles, resolution, constriction of arterioles, causes resolution, here dilatation of arterioles, compression of outflow veins. Here is constriction of arterioles. Regulation of male production, two-cell gonadotropin model. Hypothalamo-pituitary testicular axis. So hypothalamus releases gonadotropin releasing hormone, causes positive effects on anterior pituitary, which causes luteinizing hormone and follicle stimulating hormone to be released. Luteinizing hormone acts on Leydig cell, stimulatory effect produces testosterone. Follicle stimulating hormone, positive effect on Sertoli cells, causes inhibin to be released. Inhibin has negative feedback method reflects on follicle stimulating hormone, and testosterone has negative feedback mechanism on luteinizing hormone and also gonadotropin releasing hormone. Prolonged administration of testosterone causes azoospermia due to negative feedback on luteinizing hormone. Regulation of spermatogenesis. is by testosterone, follicular stimulating hormone, luteinizing hormone, and growth hormone, early division of spermatogonia. Lu- Sex hormone is testosterone. 5-alpha reductase acts on it, causes dihydrotestosterone DHT. Testosterone stimulates Wolffian duct formation of male internal genital organs, stimulates erythropoietin production, increases red blood cells. Protein anabolism increase muscle mass in males, stimulates bone growth, initiation and maintenance of spermatogenesis, stimulates synthesis of RT ABP from stimulates synthesis of ABP from Sertoli cells. Behavioral effects increase libido in men. Dihydrotestosterone is more potent, responsible for puberty-associated changes, masculinization of external genitalia, development of prostate gland, 5-alpha reductase inhibitors, finasteride, used in treatment of benign prostatic hyperplasia. So, dihydrotestosterone, more potent form of testosterone, made to dihydrotestosterone by 5-alpha reductase, works in development of prostate gland. So 5-alpha reductase inhibitors